Hello everyone, welcome to today's session. Today we will continue to cover the approaches important for working with our table of expression, which we generated last time. So last time when we met, we talked about methods for data preparation for statistical analysis. We reviewed pre-processing, mapping, and quantification. Today, we will be learning about log normal transformation, quantile normalization, principal component analysis, as well as differential gene expression as the final step of the RNA-seq processing pipeline. We are also going to introduce student t-test. So before we go into the next step of classes, next step of lessons, we will be looking at the progress like each time. And here, if you can see, like in these graphs, which represents the progress. So this program has number of online resources that will help you understand the material, and we see the progress by looking at the server access and the educational access made by you. So if I go to the page to see the progress, here I can see different humps for the graph, which shows that the educational access that you guys have been utilizing the platform and started to take a few courses. So Aditi here she has started, Soham has started, Angana has started, Nitin has started, Kamolika has been doing a few courses. She just finished Introduction to Bioinformatics. Nitin has also finished the Introduction to Bioinformatics, and it's good to see the progress. And I think in due time, you all will be able to finish the online courses. And, also remember to complete profile, and please have a username that reflects the name you would like to go by. Okay. So moving on, going back to our presentation for today. So I can see that many of you by now have been seeing the quizzes. So I would like to review two of the questions from this section and see if everything so far makes sense. If you are having any difficulties completing the material, please make use of the forum. So here I will again go back and show you the forum. So here we have we get updates. So Angana has passed the course. Very good. So let me congratulate you, Angana. So here like this, I can comment and see the progress, and you can see your own progress, and here you can write your own questions. So if you have any questions regarding anything that any problem that you've been facing, or any queries that you have related to the course or the program, you can write here. So you can write a topic. What I'm giving you an example, like what is NGS? So you can write like this, that I did not understand the data analysis that was shown in the class. Can you help? So you can write like this, and you can post so that we can see and give you answers. So it's very simple. So let's go back to the presentation again. So as I was telling you that we would be asking you some questions or to answer those questions, so that we know that we all are at the same level. So if you can answer this, which of the following are pre-processing algorithms? So last time I hope that we covered the pre-processing algorithms. So is it answer number one? Is it answer number two? Or is it answer number three or four? Can, I mean, you guys can totally write on the chat box or unmute yourself. Can you tell? Okay. So let me answer. The answer is B. PCR clean removes all the duplicate reads from raw sequencing data. The trigonometric algorithm cleans technical sequences from raw sequencing data. So that is the correct answer. Top hat and bow tie here. Top hat and bow tie are mapping algorithms. So making answer choice A and C are incorrect. DESEC and EJAR are tools for differential gene expression, not pre-processing. So that makes the answer D also incorrect. So the right answer is 2, PCR clean and trigonometric, as you can see in the image. Okay. So going to the next question. This, I think we did in some, we did explain in detail. So what types of files are used as input for RNA-seq pipeline? So what I'm asking here is that what kind of files would you use as an input? So the first answer, first multiple choice question selection would be FASTA or FASTQ files, which include DNA sequence data, or 2, SVL or URL files, or 3, RSEM or text files, or 4, Excel files. Can anyone give me an answer? Okay. Answer 1 by Dhriti. Absolutely correct, Dhriti. I mean, you're point on. So the answer is A. Yes, Angana, absolutely. Option 1. That is the correct answer. Thank you. Thank you for telling. So is it A? Is it 1? Or is it 2? Oh no, it's 1, I think. So the answer is A. You can upload files via link, which are SVL or URL bulk upload options, but the actual files analyzed are FASTA or FASTQ files that contains the DNA sequencing data. Remember that the molecule sequenced for a RNA-seq is cDNA. I think we covered it in the last session, that how the sequencing machine works and how we use cDNA, which then and then we use then it into mRNA, then it changes into transcribes into mRNA into, and then into protein, translates into mRNA and transcribes into protein. So this is what we covered last time. So also remember, the actual sequences that we are looking in a DNA is, I think everyone knows that it is ACTG, not ACUG as in RNA. So can you tell, I mean, what is the difference between FASTA, FASTQ and GTF files? I think we did cover that as well. So remember, as we covered last time, it is important to remember this, that FASTQ files provide us with reads from the sequencer. So that's how we get the reads from the sequencer. So those are FASTQ files. It's a text-based file format that stores nucleotide sequences and reads, and also quality scores for each letter. If you remember last, we did this, we saw the FASTQ files. And also FASTA is a text-based format for representing DNA and protein sequences. Typically, this is the reference file for the genome, as we also mentioned last time. The GTF file contains the genome annotation. This file will store the start and the end of each genomic element, exons, isoforms, genes. We saw that as well. So thank you again for participating. And I think if you go through the quizzes, you will be able to answer many of these questions, very simple questions, which are nothing but based out of logic. That the logic here was to understand that it is an RNA-seq pipeline, but what people usually make mistake is that thinking that it would be RNA-seq data. But you know how the machine works, that we put DNA, cDNA, and then it changes to mRNA, and then we do the sequencing. So the answer is 1. So this is the whole process that I was describing before, that via replication and then transcription and then translation, with which the central dogma. Before we get into today's material, I wanted to recap on a few concepts from the last lesson. So as we were going through the different steps. So recalling our analogy, so analysis of RNA-seq data is like a pipeline. Imagine data flowing like a river. So it's like a downstream river. It's flowing from the top to the bottom. So this is the process in bioinformatics. Downstream references the reduction of data. So reduction of data complexity. So we started with like thousands and millions of reads, and then we are coming to some important genes. So the data complexity from unstructured set of reads to a structured table of expression, and further into biologically interpretable elements like genes, gene ontology or pathways. So we are coming from like really rough or raw reads which has no meaning at all. If you see them, it's very difficult to understand what they actually mean, to a place where we can understand the meaning. So this is what you will be learning in this whole process. So it's very interesting because you see the changes in the data, you see what it is in a raw form, and how it becomes so important and informational by using these data analysis methods and technologies. That is the purpose of this program, to learn about these techniques. So just imagining, I mean, how would I do it? So if I someone asks me what does PCR clean does or Trimomatic does, I would totally do one thing, that I would remember the schematics. So the schematics is what we did in last class. So just let's go to the server for a while and just look at the pipeline. So it would help us imagine, and also it would help us to learn and remember, which is very important. So here I remember this schematic, I remember how it looks. So I also remember that pre-processing is at the beginning, where PCI clean and Trimomatic is there. So this is pre-processing. I also remember that mapping on genome has bowtie 2G, top hat 2, high sat 2, like these algorithms, which are into one logical place. So you remember like this. Isoform construction, I remember, okay, coupling is there because it is right here, because I visualize this image and then I get the answer. So I mean, to start from the basic and to develop a learning, if someone comes, like some question comes, or some colleague of yours comes and asks you that have you heard of bowtie 2 or bowtie 2G? Bowtie is a very popular algorithm which is used in many analysis. So would you be able to tell that person that what is bowtie 2? I mean, why this bowtie 2 is used? So it is used to map. This is a mapping algorithm. We know that, we learned that in the slides, in the schematics. So if you just see the schematic, you learn a lot. That's the point I'm trying to make here. that even by looking at it, you learn a lot to, tell people, you learn a lot to, write in the answer the quizzes. So coming back to the presentation, yes, we did this. We did this together. We did saw some of the algorithms, and we'll be looking into them more and more as we move into this whole, journey. So, to what we did is that our first step was to clean up those reads, then organize the reads and place them like mapping. So after that, we get a table of expression. So we cleaned it and then we aligned it, mapped it, and then we did the overlapping, and then we got a table of expression, right? That was the part we finished last time. So you have the table of expression. Once we have the table of expression, the next step is the quantification. Now we can do what's called the first downstream analysis. That that would be statistical analysis. So which kind of statistics do we need to identify what is here is signal and what is here is noise? It is important because out of 100,000 or 7,000 genes, for example, so like 100,000 genes or 7,000 genes, we need to extract some genes that are informational for our comparison between one sample. In our case, we are comparing triple negative versus ER positive, like we showed you the data set, and we will be going through it again, so nothing to worry about. Or we want to just understand like what are the variations. So those are the things that we are trying to look. But I mean, how this process is flowing from raw data to we reaching to the place where we are able to do that. So we'll be going through that. And also, I mean, if you have not understood, I mean, what we have gone we have covered so far, we will be having another session on Wednesday where we will be recapping everything. And by that time, I think many of you will be able to learn some of it. And through these interactive sessions and through these lectures, I think some of the concepts are getting cleared. But then again, we will have that recap session where we will be doing it again so that it stays with us. So moving on, I mean, for today's session, we will be covering a lot. So by not wasting much time. So you saw the workflow. And this first part here is covered in transcriptomics 1. Sorry about that. So the first part here, when we are talking about HiStat2, and we did the expression, table of expression, and then we also look into the data and see some patterns, which we will be covering today. But that's the part of T1, which is transcriptomics 1, and other steps which are like differential gene expression, annotation. Those will be part of transcriptomics 2, which we will be looking into in the next classes, in this class and the upcoming classes. So today, as a result of running the RNA-seq pipeline, we get a table of expression. And today we are going to talk about the table of expression. So in this session, we will talk about approaches that are important for working with our table of expression and preparing it for downstream analysis. So as I was telling you before, that we will be talking about transformation, normalization, PCA, factor regression or regression in general, and also we'll be elaborating more on it in the next class also, as well. So how is it like to work with a table of expression? So what we did last time is, let me just go to the server and show you real quick that how we got this table of expression. So here in home page, I will go to our favorite demo pipeline, which is the PDX model pipeline. And here I will start. So what we did last time was we started and we did perform Trimomatic, and then we did PCR clean, and then we did bowtie 2T for mapping it to the transcripts. So we did pre-processing, and then we left some of the steps because we were not looking to map into the genome, and we were primarily mapping into the transcriptome. And then we generated the table of expression from this mapping, and we got some results. So what results we got? Let's look at those results. We got results like this, and this is the file to be downloaded, which has all the results. So let's do that. Let's download this and look at it. So this will be available to all of you. I mean, you guys can simply run the same steps and get uh, these are the steps. So you start with PCR clean, Trimomatic, go to bowtie 2T, and then table of expression, and then end. And these are the results that we got. And let's just look at the table that we have downloaded, the data that we have downloaded. So here I can see a number of files. So I think it is visible to all of you. If it is not, then please let me know. So we have got here, if I don't include the temp, then we have like 1, 2, 3, 4, 8 files, and 4 files are for isoforms and 4 files are for genes. Okay. So let me, I mean, I want you to put some attention here, some focus. So here we have 4 types of file for genes: express genes not filtered, express genes FPKM, express genes FPKM not filtered. So we also talked about FPKM last time, that the output of these algorithms like RSEM and also HTSEG will give you outputs in FPKM, TPM, or RPKM. But we would take this data and further do the analysis to understand how these numbers are coming and what to do with these numbers for the next level of analysis. So expression genes not filtered are the genes which has 0 values. So we talked about the 0 values last time, that many genes have expression 0 throughout the whole sample. So from like different samples from different patients that we collected, or different mouse that we collected, that expression of that single gene throughout the samples is 0. So we don't want to use keep that, because that will have no significance in our study. So let's just open this file and show you guys how it looks. So it's a big file, 11 MB file. So that would have a lot of genes with 0 expression. So you see we have like how many genes? We have 96,698 genes with a lot of 0 expression. Now, the good thing about this platform is that it provides you with genes with filtered that has no 0 expression. So not filtered is the one which has lot of 0 expression, which we will not be using. So not filtered is something that we will not be using. The one which is filtered is that expression genes FPKM. We will be using this one because we want to use the FPKM reads, and the expression genes which are the list of genes with expression are also listed here. So if I open, you saw here like 96,000 genes, and let's see the number of genes that are without 0 expression. So let's check it out, like how many genes we have. We have here, we have 3,311 genes only. So the lot of noise or lot of insignificant genes which we were not needed were removed. So this is, as you saw, like the flow, like it's getting reduced. The water coming from the uh, mountain, and now it is becoming less and less, less and less as it's traveling. In the same way, the data which was like a lot of had a lot of noise, a lot of artifacts, and a lot of zero expression are now reduced to 3,000 genes. So this is the table. This is the FPKM table that we are of. Is it that of interest? So you can totally download it from here. And these are the files also listed here. The same files that we opened. We also have mapping statistics to understand what kind of mapping it. I mean, how good was the mapping, and all those things are covered in Transcriptomics 1, and you can look and learn more. So going back to the presentation, so this is what we did, and what we have got out of it is like when we started the analysis. Just let me. Yeah. So when we started the analysis, we had a library of unstructured data, unrecognized reads in a text file, also called FASTA or FASTQ. The output from the pipeline is called the structured data, where columns are sample names and rows are gene or isoform in ensemble format. So inside each cell, I mean, you will find quantified expression levels. So depending on the input data and quantification approach we select, these numbers are reported as, like, the quantification is FPKM or RPM or TPM or raw count unit. So this is like, we want to not look into just the raw count. We want to look at into the FPKM, TPM, or RPKM. I mean, whichever is the output. So the table contains thousands of genes and isoforms. So we need to summarize this data to explore the expression profile of interest. So we are looking at into the whole of data, and but we don't want to look into the whole of data. We want to look into those interesting genes which are differentially expressed. So one gene which is like very high expressed in a disease condition might be useful. If we can learn about that gene, we will be able to understand why maybe this disease is happening, because that gene is very highly expressed. So we are looking into few genes. That is the purpose. That is the one of the main reason to do this analysis. So the table contains lot of genes. One of the main challenge is the downstream analysis, which is what I'm explaining, is to find out specific genes or gene sets and annotate them to by their biological function. So all this jobs or opportunities, or the RNA-seq data analysis sort of things that you see, requires you to go through these processes. It requires you to do all these so that you can do some analysis which is meaningful. It's not just for the sake of doing it, but you're doing something which has some importance. So this helps us understand what expression difference can be observed in collective samples. So right now we are still, not being able to visualize the number of samples and the number of genes that are there. So if you see here, I mean, in one gene, so in for one gene, there are many samples. So this one gene has an expression level of 2.15 here. In another condition, in another sample, it is like this. So it's changing in every sample. Okay. So if you have any questions, I mean, you can go ahead and ask me right now, before we go into the next slide. But I mean, some of the basic questions here is like, what is the column name? I mean, what is the column name? I just explained it to you. What is the row name? Why does the number of numbers in this cell, what does this number in this cells corresponds to? Can anyone tell me what are these numbers? These are expressions, right? The expression level. Yeah. So what does zero means? Zero means no expression, right? That is also clear. So the column name is the gene ID. The gene IDs are there in the column, and the row has samples. Simple as that. The numbers are expression level. Zero means no expression. So if you see these numbers, I mean, it's something that I was expecting that someone would ask, that why these numbers are in decimal, 0.61, 0.23, why is like that? Can anyone tell me that? The answer is within the formula that we talked about FPKM and PPM. So the decimal is because we are dividing by the length of the gene. So all these counts are divided by the length of the gene. We have a number of reads that, I mean, number of reads cannot be decimal, right? It could be 100, 50, 20, or 81, 21, whatever, like a digit, like it will be a number, but it will not be a decimal, because if you count them, there is no half read. So it's like it's a read, right? So it is decimal because we are dividing it. We are dividing it by something. So when we divide, we get a chance of obtaining a decimal point. So that's the simplest explanation of this expression table. I hope it is clear to you now. Okay, so now the important thing about before we start looking for specific genes, as I was telling you that we want to look into specific gene, we need to look into the whole data set and understand whether it is a good data or not, whether we should proceed it with or not. What are the statistical features of this data set? So that is what something that we are trying to do here today. So to do so, we often use a box plot which shows how all the values are distributed between samples. So this is an example of a box plot. So uh, if we have multiple data sets, like if we have different samples, different conditions, in our case, it's the sample data of gene expression with multiple samples. Box plot helps compare the samples using statistical properties such as the mean, the median, and the bulk of the data. So we are trying to look into the mean and the median. For example, here, like it's a big data. So sorry about that. So I mean, in this box plot, you can see that majority of data points that are supposed to be in interquartile range are barely visible. So these are the different points. So if you see for the first condition, the median is between 0.0 and 0.2. But if we have to calculate for the whole number of all the genes, I mean all the conditions of this one gene, it is difficult to calculate that because of the outliers, because the distribution is not linear. So the outliers is are very something that is of equal importance that we have to give. So when we prepare a box plot of a typical RNA-seq data set, we can see that the gene expression levels are heavily skewed in linear scale. The majority of data points, the lower gene expression, have very low values. So this is how the whole plot, how the whole plot would look. This is the box plot, the earlier one. This is the box plot of few samples. Here there are only 1, 2, 3, 4 conditions. If I plot the number of conditions here, and if we see the expression raw reads, we will see that many of, I mean, you can see the values that are below, and there are like expansion of values, and then that's how it is going. So when you prepare a box plot, we can see the gene expression levels are heavily skewed in a linear scale. The majority of data points which are like low value between 0 to 10, and the remaining outliers, like if you see on the top, like 0 to 10, you can see a number of congested space, and on the top you can see that between 10 to 16,000 are uh, are there, which are highly expressed genes. So in this box plot, the majority of data is not visible. So these are the also the outliers. This type of distribution will infer with many of the statistical tests we plan to analyze for our expression data. So these are the problems with gene expression data when you just plot them and see them just like this. So that's why we want to do sort of different type of statistical analysis and tests to improve this data, to make it more uniform for our analysis, because we have multiple samples for one condition. And like, if you think about that gene in different in different samples is nothing but a biological replicate, right? It's the same gene in different individual or different samples, but it's the same gene. So
Hi, everyone. I think there is a technical problem on Dr. Mohit's part. So we will wait for a minute. He will be joining back again.
Hello, good morning. Maybe what we can do, it looks like Mohit is having an issue. So I wonder if we should start and go over the material without him. So let me really quickly pull up the presentation here. Okay, so today we're going to start off. Let me actually share my screen. Okay, please let me know, Bipchi, if you can see my screen.
Yes, Ilya, I can see your screen. Mohit was actually talking about box plots the last time. So he covered quite a bit of the presentation.
Okay, that's great. So I'll just try and pick up from there, and let me know. Let me know if this transition hopefully will be somewhat smooth. Okay, so was it here that he left off?
He was starting with the log scale, yes.
Okay. Okay, well, I'll try and pick up here, Mohit. If Mohit gets back, let me know. We can, definitely transfer it back over to him. But as he probably mentioned, we started with a table of expression in regular scale. That regular scale, as you can see right here on this side, really does not provide you with enough detail about the main properties of data. The properties of data that are important for a parametric test, a statistical parametric test, they assume a certain characteristic of the data. That characteristic of the data includes the mean and includes the variance. So maybe he has already mentioned this, but a regular box plot is just a way to visualize all of your data and summarize it in a simple way like this. And on this screen, what you see are several different things. For example, you can see the median, the middle of your data. You can see the interquartile range. So this is the bulk of your data. Then you can see the tails of your data. And sometimes you will have outliers. These could be conditions. So you can group, for example, multiple samples in a group and look at distribution of data by conditions, or you can look at for individual samples. So in our case, if we just do box plots of our raw data, these are individual samples. So here we have two conditions. A number of them are one condition, a number of them are another condition. Now here, if you look at this, you're primarily looking at the outliers. And the majority of your values are just on the bottom right here. So it's really impossible to even visually see those main properties of your data. You can't see the median, you can't see the mean, you can't see the variance. And this is somewhat of an example as to what happens with a parametric statistical test. It just does not have enough information to accurately determine the difference between the means of groups. So log scale transformation, as we saw, and this is the same data after log normal transformation, or after log transformation, and this is called log normal distribution, really stretches out the bulk of the data, which was previously down here, stretches it out, and now we can compare these samples much more accurately. So this log normal transformation, really all it is a natural logarithm of the original value. Sometimes you have 0 as the original value. So if you want to do this in Excel, for example, you have to do logarithm of the value plus 1, because of course that does not change the logarithmic scale. Now, if you do the histogram of an individual sample, right? So right here, we go back real quick. This is an individual sample. If you do the individual sample as a histogram in normal scale, you can see that the majority of your data is in the first bin between 0 and 70. So if you take all of your genes and all of their expression values and group them together in buckets in these different portions, you'll see that the bulk of your data is right here. And then a little bit less here, and then down here you barely have any, right? So because your data looks like this, all of your information is essentially in this first bucket. How do we stretch it out? After log normal transformation, it gets stretched out. And what does this look like? This looks like normal distribution. So that's called log normal transformation, and this would be your normal, close to normal distribution. Once you have normal distribution, that is the parameter for this parametric test. It assumes the model of the t-test, for example, of a statistical difference, assumes this normal distribution of data. Now, another useful way to be able to compare samples between themselves is to really adjust the samples between themselves using noise as a way to normalize the bulk of the data and the mean of the data. So quantile normalization, what it does is it takes the noise between samples and aligns them in such a way that the distribution between samples as well as their means are more aligned. Now, this is important, especially when we are considering multiple samples to be replicates of a condition. So if there's a condition and I have multiple samples that I'm saying represent that condition, I ultimately want that condition to be somewhat consistent across samples. And that's where I can use quantile normalization. However, I use a threshold, so I don't want to normalize everything. And so one of the questions is, what is this threshold? So if we look at the quantile normalization here, you can see that, right, so let's just quickly compare it to the previous box plot that we had here. You can see here variation in each individual sample, right? Some of them, for example, have outliers here. Others don't have outliers. After log normal transformation, we do see a lot more information, but now look inside each condition, there's a lot more consistency in the mean and a lot less variation in the outliers. That doesn't mean that the variation does not exist, but they are much more comparable. Now, they're not exactly the same. We obviously want to leave the signal part of the data. What is the signal part of the data? If we again look at normal distribution, we assume that each cell will be alive, right? So each cell is alive. It has the normal function. So therefore, you have a lot of normal gene expression. So here you can see normal gene expression. That is the bulk of your data. So the bulk of your data is going to be your vital functions. If you have a person that is diabetic and a person that is non-diabetic, you look at them, they're still living people, right? So that's not what you want to compare. The same with cells. They're still living and functioning cells. What do you want to compare? Well, you want to compare those things that are different, right? So the best way to describe something different is if you have a lot of information about those different things. And those are the significant signal. Those are the highly expressed genes that actually affect the characteristic of that cell function or of that cell condition. And you also have this technical noise. This technical noise comes from low expressed genes. They're expressed at such a level that even if you compare them between different conditions, they won't give you enough information to determine whether that is noise or that is signal. So quantile normalization uses a threshold. You can see this is right around the 5 mark. And we can use this cutoff of 5 to normalize everything underneath here and leave the significant signal as the main signal that we want to compare. As a result, after quantile normalization, you will go from something like this. This is in raw FPKM values. And this is in after quantile normalization with log normal transformation. So you have, first of all, you can see that the values here changed a lot. Here we have from 0 to 50,000. Here we have from 0 to about 10. And you can also see what the difference really is between this group of samples and this group of samples. So at this point, let me pause real quick. And first of all, Mohit, can you let me know if your internet?
Yeah, I'm here, Ilya. I can take over with you.
All right, great. Thank you, Mohit.
Thank you, Ilya. Thank you so much. So yeah, as Ilya was describing, do you have any questions for the presentation so far? Okay, so we got to understand that it is important, to remove the noisy data where, from thousands of observations, we are reducing to certain observations and we are looking into individual genes and we are comparing them. and we also compared the genes or samples with their, after doing different sort of normalization. So we start with log transform normalization, and then we do quantile normalization, which helps us to, reduce the data further and make more sense out of it. So we tested these assumptions by by plotting these data. So if I plot all the TN triple negative uh, samples and put them in a same plot, plot all of the gene expression in a same scatter plot. So if you can see, the samples were, if the samples are identical, as I was telling you before, their level of expression should be aligned perfectly along the diagonal red line, as you can see here. So if they are correlated, the value should be stretched out along this line, like properly in a single line. So now that we prepared the raw data, let's see how we can learn, about the whole data set, or learn about the data and the properties of the data. So we can't see if the genes have, comparable expression level here. We just can see that most of the data is concentrated around the low values here and scattered all around. So it is a, challenging task because of the outliers which we transform. And now we will try to understand that how data is being transformed from thousands of samples to few, by looking into the variation. So in RNA-seq data analysis, one of the key aspects that we're looking at is the variability or the variation in data. So for that, we would be using, as I was telling you before, principal component analysis. So let me just go to the presentation. Yes. So principal component analysis. To study how gene expression can be used to measure, variability between samples, as I was telling you, we can use dimensionality reduction technique that is quite often used in the RNA-seq data, and it's a very popular technique in bioinformatics as well. This analysis method will help us visualize the multidimensional data set. So I will be explaining what I'm talking about in a scatter plot, and see how gene expression pattern shows about the data. So talking about about the dimensionality. So if we have 2 genes and we have 8 samples, and this is the gene expression, we can totally plot it into a 2-dimensional plot with x and y axis. So the genes are dimensions and the samples are objects, as you can see, and you can plot it in a 2-dimensional graph. And if you have 3 genes and 8 samples, you can still plot it on a 3-dimensional graph, right? That is quite easy. We can do it in Excel with small data like this, and we can see that it's easy to plot 3 genes and multiple samples in a 3-dimensional graph. But in our data set, we don't have 3 genes. If you remember, I showed you the zero expression file, which had like 96,000 genes, and then I showed you the file without the zero expression, and we still saw like more than 3,000 genes. So 3,000 genes, and we want to see how the data looks like. How would we do that? It is not possible to humanly plot this into a multi-dimensional graph and see it. That's not possible for us, but that is possible for the machine learning technique called PCA, which can plot multi-dimensional data, and we can see the visualization. So how does PCA work? So in PCA, we were talking about the variance. So here in the first graph, you can see it has low variance, right? In the second graph, you can see it has high variance. So why low variance? Because the data points are close together, and so that shows low variability. Here the data is more spread out, which shows high variability. So in principal component analysis, what we do is that we try to find out the variance between the data, so how they are different, and then we plot it using a mathematical model, which we will be discussing more and more in the machine learning class. So we use vectors and we plot them in a two-dimensional graph to see the variance, to understand the variance. So components which are generated through PCA are plotted to which explains the variance, and which we will be doing in our hands-on exercise today as well. So last time, as I showed you this before, that we did the pipeline where we started from cleaning, and then we did the mapping onto the transcripts, and then we generated the RSM table, and this the output of this pipeline yielded us our table of expression, which in which we took the FPKM values, and then we looked into the expression and the read counts there for different number of samples. And then today we learned about different steps of statistical analysis like log transformation and quantile normalization. So this is the file which we said that we will be working for our downstream analysis. So in a PCA, let us just go and download these files on the platform, which we already downloaded, which I already showed you that these are the file, and this is the file of our interest, that expression of genes in FPKM. So also this is not filtered, so it has limited number of genes which are of interest. So let us download this file only, and we will be using this file in looking into the data further, and we will look how PCA is able to give us some visual idea about how our data set is. So today we are going to learn how to build a new pipeline, which is in which you can upload data. All these times we were looking at the demo pipeline, RNA-seq PDX project, and we also gave you an overview of how to build your own pipeline, but obviously which will come in the next step. But today we are going to use the utilities, and we will learn how to do all the things that we talked about today, like quantile normalization, log transformation, and also PCA in one step. So here, the first step is to upload the file. You have several options here, but to do PCA and normalization, different kind of normalization, we would like to, the server would ask you to upload the file that has been generated. So we have downloaded that file. So let's just, add that file. So let me just start over again. So here in the home section, we did the analysis here, and we ran the pipeline and downloaded the data. After that, I took the FPKM file, and I want to do some normalization. Okay. So I will come here and I will press this button to upload file. Here it's asking me to please upload file. Here I will add file. I'll go to the downloads, and okay, I have already downloaded them on this computer as well. So here are the files. So expressed genes FPKM, open. Okay, so you can see here the file is uploaded 100%. We can press now. We also discussed in the quiz about these, and we will be discussing more in the coming days about different types of files and uploads. But today, like as we said, we have uploaded it. Now we can create a new pipeline. So let's press start. Okay. And we wanted to do quantile normalization, right? And I think Ilya also showed us that why we need to do quantile normalization and why a threshold is important. Because you saw the data hump and you realized that most of the data lies in the normal gene functions. All those data with normal gene functions lies in a certain area. So we want to do first transform table to logarithmic scale. Yes, we want to transform the scale, table to logarithmic scale, because that's the transformation we want. And also we want to put the threshold to 5 based on the RNA-seq data that we have and save. Next option is PCA. So we can do quantile normalization with that data and also do a PCA with that data. So here we will be doing by max 25%, and we will be again doing PCA, and where we will be explaining the parameters in more detail. But here these are the default parameters, and this is to run this pipeline, we will be using the default parameters by only changing by max 25%, which we will be discussing. So we are considering 25% of the data. So that's why by 25, max 25%, and then save, and then we will end the pipeline. So once you end the pipeline here, like in the demo pipeline, it was like, it automatically runs. But when I save the pipeline, it will ask for a name to this pipeline, because this is my pipeline. This is not a demo pipeline. Save. So it's asking for a name. So what are we doing here? So we are working on PDX, and we are doing quantile normalization, and we are also doing PCA. I'm writing like this so that I remember what I did. So PCA, save, and then it asks me whether I want to run the pipeline or not. So it is again important, once you have created the pipeline and did everything, you hit the button run pipeline. Once you hit the button run pipeline, the pipeline starts to run, and you can confirm that by looking at here, because here it shows that 1% started, and now you want to see where your pipeline is, where you can see all your pipelines or the one that you have started. So there is an interface called my pipeline. You can go here, click, and you will see that this pipeline is processing right now, and you'll get update here as it gets finished. But I have already done this pipeline, so let's look at the result of this pipeline. So it's the same pipeline. We did quantile normalization, PCA, and then we finished the pipeline. So if you see here the output result, these are the two results, and this is the zip file which has both the files. So we have a file called normalized threshold 5 FPKM, and also we have a PCA result file with threshold 5. So let's look at the threshold 5 was generated after quantile normalization, and then there was a PCA done on it. So let's download these files and look at what is there in them. So these are the two files. So let me go back to the presentation and describe you more about it. So we went through these steps, and we changed the threshold, and then we changed the percentage, and we got a result like this, which explains the variance in the data. So we have, we will be, again, I'm telling you, we will be describing more in our upcoming classes, but here to understand that we have captured 21% and 9.14% of the, like 30% of the data we have captured using the PCA. And in the PCA, we will get like different PC1, PC2, and we can plot this and see how the data is, looking. So, I mean, in here, the pipeline which we built showed us data in 2 columns, this file. So let me just open it so that you guys get a visual understanding. So this is the PCA result. So open it in Excel. So here you can see the variability is different for different PCs. So PC1 and PC2 captures the most variability. And also here we have different samples, and then we plot this and we get a graph like this, which shows us the distinction between the triple negative and the ER positive. So you can see here, those are separated quite well with one outlier. So notice that we can see distinct samples, distinct difference between the samples. That is what I'm coming at again and again, that we can see that the triple negative and estrogen receptor positive samples in the scatter plot, and we can see that they are being separated. So that is what PCA gives us. So to give you more an example that you can maybe correlate more with is this. So it's like, a photographer is trying to capture a picture with a lot of people, and he's looking for the best angle to capture the picture with everyone in it. So the person is trying to capture a photograph, and is trying to maximize the variance. He's trying to capture all the variability that he can see in the picture. So that is an example of PCA. Why we use PCA? We use PCA to explore data and to visualize it. What all consideration we have to make before doing a PCA is that data is normalized, data is filtered, outliers, there are less outliers, and then we will be able to interpret our PCA. So today, we covered what is there in Transcriptomics 1. So I would like to ask you a few things. So I think the conclusion and the review section, as you can see here in Transcriptomics 1, has a video which explains this very well. So you will benefit from watching the video, and also you will benefit by taking this section and reviewing and going through this section, which is really well done well. So pay attention to the bullet points at the end as you will finish this course. And also, please take some time to submit the course feedback, because your opinion truly matters to us. A lot of work has gone into building this course and developing this content, and it is very important for us, so that we understand that what you liked, what you think can help us, what changes you can think can help us improve. So please go through this. And if many of you are comfortable doing PCA or want to understand more of PCA after trying PCA, you should consider taking this course on PCA. So PCA in R, which is available on our Bioinfo platform. So let me share the link with you. Deepsha, can you share the link, please? So thank you. Yeah, so today we covered a bit, and today we looked into the gene expression. Today we looked into the table of expression. Today we did some normalization, and also we looked into PCA and how does that PCA helps us in visualizing the data. So I think this was helpful for all of you to start doing something on the platform. And this is well described in, as I was telling you, everything that I've said and that everything that we went through are well described in our courses, Transcriptomics 1 and then Transcriptomics 2. So we finished Transcriptomics 1 and we did some of the Transcriptomics 2, and which we will be continuing in the next class on Wednesday, that we have additional workshop online workshop, which would be a hands-on session where we actually would like to share screen with you, like so that you can share your own screen, and go through these steps together. So we'll be trying to do that. We'll be trying to help you understand what we have covered so far. If you have any questions, we'll be happy to take those questions during that session. And also we'll be covering some of the steps, next steps, which would be the differential gene expression, t-test, and regression analysis. But it would be very important for you also to go through some of it, because everything is there in the courses. And I'm really happy to see the progress also. So many of you are doing really well. So would like to conclude today's session, but before that, I mean, if we have any questions, please go ahead and ask, so that we can have a clear understanding. Okay, Angana, do you want to unmute? I mean, is it something that you think that you could not understand today, or what are your thoughts? Or Dheerti, anyone? I mean, Pankaja, Soham.
Hi, sir.
Yes, Soham.
Yes, just one thing that I was announcing is that about the Amity participants, we have created a WhatsApp group for the doubt clearing classes, and also for those who are facing any issue with exploring the educational platform and the server platform. So anybody is facing any issue, then he or she can ask questions also there. I mean, outside also, outside this Zoom link.
Absolutely, and thank you, Soham, for bringing that up. I mean, yes, I mean, any of you having any issues while going through the content, understanding any of the things that we talked. I mean, many of the things are new to you, and we understand that, and it would be no problem for us to keep doing it, keep telling you that this is how it works. But I mean, Soham is there and Sarah is there, so we also have this WhatsApp group. I mean, you can ask your questions and you can consult with them. They can show you, they will be able to show you it on their own computer or on your own laptop in MIT University, Kolkata. So you can also have that, demonstration with them. So Soham can demonstrate you all the things that we have covered, and he can show you. So if you have any issues related to that and regarding that, and you can contact Soham and Sarah. And if you are in IG, you can get in touch with Chandrani, as Deepsha is writing in the chat. So for today, if we don't have any questions, I would like to conclude this session, and thank you for joining. And we'll be hoping that you will be joining us for our next session, which is the continuation of this session, where we will be exploring more of differential gene expression and the downstream analysis associated with it. So thank you everyone for joining today's meeting, and have a nice weekend.